Hello friends, in this video we're going to be studying the nucleic acids. Now, the nucleic acids are biopolymers made up of repeating units of monomers called nucleotides. The nucleic acids are biopolymers made up of repeating units of monomers called nucleotides. So what we are trying to say now is that they are biopolymers. So bio there would mean that they are synthesized within the body, and like I said, the polymers are structures which are made up of repeating units of monomers, and in this case the monomers which form the nucleic acids are called nucleotides. So we can therefore say that the nucleotides are the building block upon which the nucleic acids are formed. There are two types of nucleic acids. The first would be the ribonucleic acid, and the second would be the deoxyribonucleic acid. The ribonucleic acid you commonly know as the RNA, and the deoxyribonucleic acid you commonly know as the DNA. These nucleic acids perform a variety of functions for us, such as the holding now of our genetic information. So they hold or they contain our genetic information. Other functions would include the production of different types of proteins needed in the body, amongst others. We're going to be discussing the functions in subsequent videos. Now, we said that the nucleic acid now are formed from repeating units of nucleotides. So it is important that we study these nucleotides which form our nucleic acids. The nucleotides. Now, the nucleotides are structures which are formed from three fundamental components. The first is the pentose sugar, the second is the phosphate group, and the third is the nitrogenous base. Let's study each of these components one after the other. The pentose sugar now. The pentose sugar, like the name implies, pent would mean a 5-carbon atom sugar. A 5-carbon atom sugar. So these 5-carbon atom sugars are called pentoses. For our studies on the nucleic acids, there are two important examples of pentoses that we must know. The first now is the ribose, and the second is the deoxyribose. Here are the structures of the two important pentose sugars that we need for our studies on nucleic acids. The first is the ribose, and the second is the deoxyribose. The first is the ribose, and the second is the deoxyribose. The ribose sugar is the sugar that makes up our RNA. So the ribose sugar is the sugar that forms the structure of our RNA. While the deoxyribose sugar is the sugar that forms the structure of our DNA. Now, what is the difference between these two sugars? Notice now that these carbon atoms are numbered in a clockwise direction, starting from the carbon number 1 towards the right and moving down to carbon number 5, which is outside the ring, Now, each carbon atom has different functional groups attached to it. Now, for example, we have the hydroxy group and the hydrogen atom. The major difference now between the ribose and the deoxyribose occurs at the carbon number 2. Notice now that at the carbon number 2 of the ribose, we have both hydrogen atom and an hydroxy group at the carbon number 2. But at the carbon number 2 of the deoxyribose, we have two hydrogen atoms. What occurred there was that there was the loss of an oxygen atom at the carbon number 2. Thus the name deoxyribose thus the name deoxyribose, because there is the loss of an oxygen atom at the carbon number 2. The next group we are going to study is the phosphate group. The phosphate group. The phosphate group consists of a phosphorus atom to which 4 oxygen atoms are attached, which are negatively charged. These oxygen atoms are negatively charged. Now, due to that, the phosphate group is a highly polar group due to the fact that it is combined to 4 oxygen atoms which are highly ionized. As a result, it gives it a highly polarized state. These oxygen atoms which are highly ionized impact a negative charge to the phosphate group. That's my time, guys. So in the next video, we're going to be studying the last component of the nucleotide, known as the nitrogenous bases. We're going to be studying how all these components are combined to form our DNA and our RNA. Thank you so much for watching. See you in the next video.